Hi, micro folks. These are our lab videos for spring 2020 when the campus was closed because of the coronavirus pandemic. This is the second video for Lab Manual Chapter 10. The first video was kind of an introduction to microbial growth media. So for this second video, folks, we're going to be doing the sampling airborne microbes using different microbial media. In the spring 2020 lab manual, this starts on page 115 and goes to page 116 where there's tables to record your results. So I'll show you how we would set it up. I'll actually run the experiment and then I'll come in this weekend to do the colony counts. Because what we're going to be doing is seeing how many microbes land on the surface of our agar plates, how many microbes can grow and form visible colonies. So we'll count total number of colonies to reflect the number of microbes in the air that landed on the surface of the plate. And then we'll be looking at different types of colony morphologies, the different sizes, colors, shapes, the different patterns of borders, the different textures of the surface of the colonies. And the reason for this is that we presume that different microbes will form different types of colonies. So by counting the total number of different colony varieties, we get an idea of the variety of microbes that are going on our plates. So the experiment is set up to compare two different types of media, tryptic soy agar, which you might remember is a, whoops, I can't do this. Okay, let's do it like that. As a all-purpose medium, it should support the growth of a wide range of environmental bacteria and fungi. And then the other medium we're going to test is the Sabrod's dextrose medium. And you'll recall we said this is selective for fungi. It has a high dextrose glucose content, which will inhibit a lot of environmental bacteria. And also it's been acidified. The pH has been lowered, and that too will help inhibit a lot of environmental bacteria. But our environmental fungi should be able to grow on this. So we're going to select for the growth of fungi. So for example, molds and yeasts. We have set up the experiment so that we're going to expose one set of plates. Well, actually, there's 3 time exposures. One set of plates, we will leave the lids on. That's the 0-minute exposure. That's our negative control to make sure our medium was sterile to start with, right? There was no contamination. And then a second set of plates, we'll take the lids off for 30 minutes, and then the third set of plates will take the lids off for 60 minutes. So labeling is really important, folks. So for my TSA plates, I have a TSA 0-minute exposure. That's my negative control. So I'm going to leave the lid on. I've got my TSA 30 plate, so I'm going to take the lid off and expose the surface of the agar to airborne microbes for 30 minutes. And then I have my TSA 60-minute plate. So the surface of the plate will be exposed to airborne microbes for 60 minutes. And then we're going to repeat it with our Sabrod's dextrose medium. So we'll have a 0-minute control, negative control. We'll have a SAB 30-minute exposure and a SAB 60-minute exposure. Okay. So I'm not sure if you can see this, but I've got all 6 of my plates face up on my bench, and if we were doing this as a team, one of your colleagues would be the timer. So I've got my timer right here, and I've got it set for 30 minutes. So when I first start the timer, like right now, I'm going to take the lids off the 30-minute plates, and I'm going to take the lids off the 60-minute plates. Okay? And then we'll actually do this experiment. Like, we'll actually let it run right now. So when the timer goes off at 30 minutes, I would put the lids back on the 30-minute plate so they're no longer being exposed. And then I'd reset the timer for another 30 minutes so that my 60-minute plates have a total exposure of 60 minutes to the airborne microbes. So we'll just pretend, folks, that we finished our experiment here. So what we would do, we'd have the lids now on all of the plates, the 30-minute plates, the 60-minute plates. We would take the lids of the plate to the bottom on all the plates, and then we're going to invert them to incubate them. And we're going to incubate at 30 degrees Celsius. Since we're trying to grow environmental microbes, in general, they're happier growing at lower temperatures than, say, human body temperature. So we would incubate at 30 degrees Celsius. In 48 hours, so today's Friday, so I'll be checking the plates on Sunday. We'll record what they look like at 48 hours, and then we're going to leave them for another week at room temperature. I guess I'll take them home with me, right? Because I can't come back into the lab. I'll keep them in our garage, and we'll see what they look like after. That would be, we'll check them a week from today. So we'll check again the plates at 7 days. And again, what we're looking for is total number of colonies that represents the microbes from the air that landed and can grow. And then we're also going to look for the total number of different colonies, the variety of colonies, that again represents the variety of microbes that can grow. So I'm going to take the lids back off because I actually do want to run this experiment. And then, folks, just to clear this, not to clear it up, but just like to finish up the little video here, I just wanted to show you some plates that Carmen and Val made, just to give you an idea of how different types of microbes will form different colonies. This is a beautiful Serratia marcescens. It has this beautiful brick red colony. This is a cousin of E. coli. It's one of the Enterobacteriaceae. And then here's good old E. coli. Oops, that's kind of hard to see. Let's see if I put this in front. Okay. Oh, okay. I'm not sure if you can see that, but you can see that E. coli doesn't have that red pigment, right? The colonies maybe look pretty similar otherwise. In contrast, folks, if I grab, this is a gram-positive cousin of Bacillus anthracis. Okay, this is Bacillus cereus. And you can see, or maybe you can't see. Wow, that isn't too good, is it? Okay, not sure if you can see this, folks, but you can see the colonies. They have a different size, shape. They're not pigmented. If we could see this better up close, we would see that the surface of the colony is relatively rough. I think what I will do, folks, is when we do the follow-up video on the airborne plates, since actually showing you different plates with different colonies isn't working, what I'll probably do is use the photo atlas, or maybe I'll take some of the photo atlas photos of different colonies and incorporate them into a PowerPoint with a voiceover that we'll do for chapter 10. Okay, folks, so we'll finish this little video. There'll be another video to show you the results. Okay, and then let me see here. Part 2 of Chapter 10, Microbial Growth, is looking at different selective and differential media. So we've already introduced the tryptic soy agar TSA plates. We've introduced the selective for fungi, Sabarodes dextrose medium. So then what we'll do on the next video is we'll go into more detail on our selective and or differential medium. We'll be looking at blood agar. We'll be looking at McConkey's agar. And we'll also be looking at mannitol salt agar. And again, since it's kind of hard to see here with this particular camera, probably what I'll do is use a voiceover and a PowerPoint so you can actually see nice examples of these plates with different microbes growing on them. Okay, folks, so we'll close this one for now.